What if I told you that buried beneath the Earth's surface, locked inside ancient rock, encased in ice, or sitting in the dusty drawers of university museums, there are bones, skulls, and fossils that scientists cannot explain. Not metaphorically. Not, we haven't studied them enough. I mean they have run every test available. DNA sequencing. Isotope analysis. CT scanning. Electron microscopy. And still, nothing. No match. No known family. No ancestor on the evolutionary tree that makes sense. These are creatures that existed on this planet. We have their physical remains. We can touch them, measure them, date them. And yet, we cannot tell you what they were. Tonight, we're going over 10 of these cases. 10 sets of remains that have genuinely stumped the scientific community. Not fringe researchers. Not YouTube theorists. Peer-reviewed institutions. National museums. Universities with centuries of credibility. Some of these were found in remote deserts. Some came from the ocean floor. Some were discovered by accident, by children playing, by construction workers digging foundations, by farmers plowing fields. And every single one of them has forced scientists to say 3 words that scientists deeply hate saying: we don't know. Let's start that in 1958, a fossil collector named Francis Tully was digging through the Carboniferous shale deposits of Mason Creek in Illinois. A site famous for exceptional preservation. What he pulled out of the rock that day had never been seen before. The creature had a long, torpedo-shaped body. At one end, it had a claw-like appendage with tiny teeth. At the other end, it had a tail fin like a fish. And running across the top of its body, horizontally, was a rigid bar with 2 eyes on the ends, like a hammerhead shark's head, but attached to the body differently from anything known. He brought it to the Field Museum in Chicago. The scientists there named it Tullimonstrum gregarium, the Tully monster. Why it cannot be classified? Here is the core problem. For over 50 years, nobody could agree on what kingdom of life this animal belonged to. Was it a vertebrate, an animal with a backbone, like fish and amphibians and humans? Or was it an invertebrate, an animal without one, like worms and mollusks and insects? In 2016, a teen from Yale published a landmark paper in Nature claiming the Tully monster was actually a vertebrate, specifically, a primitive jawless fish, like a lamprey. The evidence? The creature appeared to have a structure that resembled a notochord, the precursor to a spine. 2 years later, in 2018, a separate team from the University of Cambridge looked at over 1,000 specimens and completely contradicted this. Their analysis found no vertebrate characteristics whatsoever. They concluded it was an invertebrate, and a deeply unusual one with no clear relatives. Then in 2020, another paper suggested something else entirely. Right now, as you watch this, the Tully monster is formally classified as an animal of uncertain systematic position. That is the scientific way of saying, we have no idea what it is. It lived 307 million years ago. We have thousands of its fossils. And we cannot place it on the tree of life. Why this matters? The significance here isn't just the weirdness of the creature. It's what this tells us about life on Earth. If an animal can exist in large enough numbers to leave thousands of fossils, and we still cannot determine its basic body plan or evolutionary lineage, what does that say about how much of ancient life we actually understand? Scientists use something called the Cambrian explosion to explain why life diversified so rapidly 540 million years ago. But cases like the Tully monster suggest that even in much more recent periods, 300 million years ago, life was producing body plans so unusual that we have no category for them. In 1909, the paleontologist Charles Doolittle Walcott discovered one of the most important fossil sites in the history of science, the Burgess Shale, in what is now Yoho National Park in British Columbia, Canada. This site preserves creatures from the Cambrian explosion with extraordinary detail: soft tissue, internal organs, eyes, guts. Creatures that would normally leave no trace are here in stunning resolution. Among the many bizarre organisms Walcott found was one so strange that when the great paleontologist Simon Conway Morris redescribed it in 1977, he named it Hallucigenia sparsa, because it looked like a hallucination. What it looked like? The creature was worm-like in body. But coming off its back were 7 pairs of rigid spines, like stilts. And coming off its belly were 14 pairs of soft, clawed tentacles. When Conway Morris first reconstructed it, he made a logical error that would stand uncorrected for over a decade. He put it upside down. What he thought were tentacles were actually legs. What he thought were legs were actually spines on its back. But even the corrected reconstruction, made in the 1990s, didn't solve the real mystery. What was this thing related to? The classification problem. The initial assumption was that Hallucigenia was a completely unique lineage with no living relatives, a dead branch on the tree of life. Then in 2015, a paper in Nature from the University of Cambridge proposed a revolutionary connection. By analyzing the structure of the claws on Hallucigenia's legs under electron microscopes, they found a layered, nested structure, one that matched the claws found in a group of modern animals called velvet worms, or onychophrons. This suggested Hallucigenia might be an early relative of velvet worms, and possibly even an ancestor of the whole group that includes velvet worms and arthropods, which means insects, spiders, crabs, and everything with an exoskeleton. But here's what was not resolved: even if Hallucigenia is accepted as a stem group ecdysozoan, the technical term for the broader group, its exact body plan, with those back spines, remains unexplained by any known developmental biology. No modern animal grows spines like that from its dorsal surface as a primary feature. No known embryological pathway produces that pattern. The connection to velvet worms might place it on the tree of life, but it doesn't explain how it worked as an organism. The head problem. And then in 2015, the same team discovered something that had been invisible for a century: Hallucigenia had a head. This might sound obvious. But for 106 years, nobody could identify which end of the creature was the front. The original specimen in London's Natural History Museum had a blob at one end that everyone assumed was a head. It wasn't. Analysis revealed it was a stain in the rock, possibly decayed internal organs. The actual head was at the other end. It had a circular mouth with concentric rings of teeth. And a pair of eyes that were simple but undeniable. The mouth's architecture, rings within rings of teeth, has no clear parallel in any living or fossil animal. So we now know slightly more about Hallucigenia. We know which end is the head. And we're still left with an organism whose body plan, spinal architecture, and oral structure have no adequate explanation: the creature that made scientists laugh. In 1972, Simon Conway Morris, the same paleontologist who later worked on Hallucigenia, presented a reconstruction of another Burgess shale organism to the annual symposium of the Paleontological Association in London. When the image went up on the screen, the audience laughed. That's not a metaphor. They actually laughed. In the middle of a scientific conference. The creature was Opabinia regalis, and the reconstruction showed an animal so strange that the assembled experts' first instinct was that it must be wrong. What it looked like. Opabinia had 5 eyes, 3 in a row across the top of its head, and 1 on each side. It had a long, flexible proboscis, a tube extending from the front of its head, that ended in a claw facing backward. The presumed function was that it scooped food backward toward a mouth that faced downward and backward on the underside of the head. Its body was segmented with a series of lateral lobes, flaps running along each side. That may have functioned as gills, as swimming paddles, or both. The tail had 3 pairs of blade-like fins. Nothing alive today looks like this. Nothing in the fossil record looks precisely like this. The classification debate. Opabinia was initially placed in a completely separate category, a unique lineage with no relatives. Then in the 1990s, with the broader revision of Cambrian paleontology led by Conway Morris and the Harvard paleontologist Stephen Jay Gould, it was proposed as a stem arthropod, something close to the common ancestor of arthropods. Something close to the common ancestor of arthropods, before the arthropod body plan was fully assembled. This made logical sense. Arthropods have segmented bodies, appendages, compound eyes. Opabinia has some of these features in primitive forms. But here is what this classification doesn't explain: the 5 eyes. Arthropods have compound eyes, many facets in one structure, or simple eyes. Some have both. But no arthropod, living or extinct, has 5 separate eyes arranged in the pattern Opabinia has. The developmental pathway to produce that would have to be something we've never documented. And the proboscis. No arthropod, not a single species among the millions known, has a frontal proboscis ending in a backward-facing claw. Not even close. So Opabinia might be a stem arthropod. Or it might be something that independently evolved some arthropod-like features. The 5-eye configuration and the proboscis remain deeply, genuinely unexplained. Gould wrote about Opabinia in his famous 1989 book, calling it the most theatrical moment in the modern revision of the Burgess Shale. Scientists laughed. Then they spent decades being unable to agree on what it was. A finger bone in Siberia. In 2008, archaeologists were excavating Denisova Cave in the Altai Mountains of Siberia, a site that had been yielding tools and artifacts for years. In a layer dated to approximately 41,000 years ago, they found a bone fragment. A small piece of the distal phalanx, the tip of a finger, from what appeared to be a young female. The fragment was too small and too fragmented for traditional morphological analysis. They couldn't study its shape. So they sent it for DNA sequencing to the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany, where the paleogeneticist Svante Pääbo, who would later win the Nobel Prize, was leading the field. What came back stunned everyone. The DNA results. The mitochondrial DNA from the finger bone was unlike anything in the database. It didn't match modern humans. It didn't match Neanderthals. When they sequenced it and compared it to the known family tree of the genus Homo, the divergence was so significant that the last common ancestor was estimated to have lived approximately 1 million years ago. Neanderthals and modern humans share a common ancestor roughly 600,000 years ago. Whatever left this finger bone had split off from our lineage much earlier. They named the new human species, and it is a species, formally accepted, the Denisovans, after the cave. The physical problem. Here is the extraordinary scientific situation. We have extensive DNA from the Denisovans. We know their genome. We know they interbred with both Neanderthals and modern humans. Modern Melanesians, Aboriginal Australians, and some Southeast Asian populations carry 4 to 6% Denisovan DNA in their genomes today. But we have almost no physical remains. A finger bone. A wisdom tooth. A toe bone. A fragment of skull. A jawbone discovered in Tibet, which showed Denisovans lived at extreme altitude, which is itself remarkable. From these fragments, we cannot reconstruct what they looked like beyond rough estimates. Analysis of their genome suggests they may have had dark skin, dark hair, and dark eyes. They were likely large-bodied, based on the jawbone. That's essentially it. We know a human species existed across a massive swath of Asia for hundreds of thousands of years. We know they were genetically distinct enough to be a separate species. We know their DNA still lives in modern humans on multiple continents. And we cannot tell you what they looked like. We cannot tell you how they built shelters, how they communicated, what their social structure was, or what caused their extinction. The physical remains, the bones, are so few and so fragmentary that they match no known morphological profile. You cannot pick the Denisovan out of a lineup of ancient human skulls because we don't have a skull. What the genome reveals and cannot reveal. Genome analysis can tell us certain things. It can tell us about population bottlenecks, interbreeding events, migration patterns, certain physical traits linked to genes we understand. But the Denisovan genome also contains large sections that are not found in modern humans or Neanderthals, and whose function is unknown. These genomic regions are referred to as Denisovan-specific sequences. Some have been passed on to modern humans and appear to have functional significance, including one variant that helps Tibetan populations survive at high altitude, but many others have no known function or analog. Svante Pääbo, in his Nobel lecture in 2022, called the Denisovans a ghost population that we know genetically but cannot see physically, the creature that was three animals. This is one of the most remarkable cases of misidentification in the history of paleontology, not because scientists were careless, but because the animal was so fragmented in the fossil record that its body parts were mistaken for three separate organisms for nearly a century. In 1892, a paleontologist named Joseph Whitteaves found a fossil in the Burgess Shale and described it as the tail of a shrimp. The tail was actually a frontal appendage, an arm, of a much larger creature. But they didn't know that. Separately, a disc-shaped fossil was found and described as a jellyfish species. A circular structure was described as another invertebrate. All three were considered separate animals. In 1985, Harry Whittington and his student Derek Briggs reanalyzed the Burgess Shale material and realized something, that a shrimp tail, that a jellyfish, and the circular structure were all parts of the same animal. What Anomalocaris actually was. Anomalocaris canadensis, which means, abnormal shrimp from Canada, a name that stuck even after it was understood not to be a shrimp at all, was a predator. Roughly 1 meter in length, making it the largest known animal of the Cambrian period, by a significant margin. It had large compound eyes, among the most sophisticated visual systems found in Cambrian fossils. It had two large, segmented frontal appendages that functioned as arms. Its circular mouth had a ring of hardened plates. Not exactly teeth, but tooth-like structures, that opened and closed like an aperture on a camera. It swam using lateral flaps along its body, similar in function to the fins of a ray. The classification and body plan problem. Anomalocaris is generally placed in the group Radiodonta, a grouping of Cambrian predators with this distinctive frontal appendage and circular mouth configuration. But Radioiodontans don't fit neatly into any modern phylum. The current consensus is that they are stem arthropods, early lineages on the branch that would eventually lead to insects, spiders, and crustaceans. The frontal appendages of Anomalocaris are considered potentially homologous to the frontal appendages of modern arthropods. But the circular mouth structure, the oral cone, has no clear homologue in any living or well-understood fossil arthropod. Its mechanics are still debated. Some analyses suggest it couldn't have processed hard prey like trilobites. Others, using physical modeling, suggest it could. Even more puzzling, multiple species of Anomalocaris and its relatives have now been found worldwide, from the Cambrian through the Devonian period, a time span of roughly 100 million years. They were successful predators for an extraordinarily long time. And yet, they left no clear descendants. The lineage simply ends. A dominant predator, globally distributed, evolutionarily successful for 100 million years, with no descendants and a body plan that doesn't fully connect to any living group. A photograph from the ocean floor. In 1964, the American research vessel USNS Eltonin was conducting a photographic survey of the ocean floor southwest of Cape Horn, Chile, at a depth of approximately 3,900 meters. The camera lowered to the bottom captured a single image that would become one of the most persistently debated photographs in the history of oceanography. The image shows what appears to be a structured object rising from the seafloor. It has a central stalk. Radiating from the stalk, in precise geometric intervals, are horizontal branches. From each branch, smaller branches extend. And from those, small orbs or knobs appear at the ends. The symmetry is striking. The geometry is regular. It rises approximately 60 centimeters from the seafloor. The initial explanation, and its problems. When the image was first widely distributed in the 1960s, the immediate scientific response was that it appeared artificial, that its regularity and geometric precision didn't look biological. This interpretation was seized upon by UFO researchers and alternative historians who proposed it was an alien artifact or an ancient antenna of unknown origin. This framing stuck in popular culture and led to the object being called the Eltonin antenna, a name that persists today. The mainstream scientific community countered by identifying it as a species of carnivorous sponge, a hexactinellid sponge, also called glass sponges, called Cladorhiza or a related genus. These sponges do have branching structures and can grow in regular patterns. But here is what is not fully resolved. The image, while low resolution, shows a geometric precision in the branching pattern that exceeds what has been well documented in known sponge species. The regular radial symmetry at each level, where branches appear to divide at consistent angles and consistent lengths, has not been matched in subsequent examination of known hexactinellid sponge specimens from that depth and that region. No follow-up specimen. The more significant problem, despite the Southern Ocean being extensively studied since 1964, no specimen matching the precise morphology in the Eltonin photograph has been collected, named, and formally described. Multiple researchers have pointed to similar-looking sponges. None of them is an exact match. The photograph remains the only documentation of whatever that object is. Is it a sponge? Almost certainly something biological. Is it a known, classified species? That is genuinely unresolved. The Eltonin image shows an organism, or a structure, at the bottom of the ocean that has not been formally identified to species level in over 60 years of subsequent oceanographic work. Given that estimates suggest over 80% of deep ocean species remain undiscovered, the explanation, it's some kind of sponge we haven't catalogued yet, is entirely plausible, and also means it's a creature whose remains, or whose living form, don't match any known species on Earth, the Ediacaran biota. Before the Cambrian explosion, before the great diversification of animal life, there was another period of complex multicellular life. The Ediacaran, running roughly from 635 to 541 million years ago. The organisms of this period are preserved primarily as impressions in rock, soft-bodied creatures with no hard parts, leaving only their outlines and surface textures in ancient seafloor sediments. One of the most common and most studied Ediacaran organisms is Dickinsonia. First described in 1947 from specimens found in South Australia, Dickinsonia has since been found on multiple continents, making it one of the most globally widespread organisms of its era. It looks like a ribbed oval or ellipse. It has no head, no tail, no obvious front or back. In early analysis, it appears to have been made of a series of isomers, repeated units, running along its length. And for 70 years, nobody could agree on what kind of life it was. The classification war. The debate over Dickinsonia's classification was one of the longest-running arguments in paleontology. Was it a plant? Some researchers said yes, its lack of obvious animal features and its sessile existence suggested it might have been photosynthetic. Was it a lichen? A fungus-algae composite? This was proposed multiple times. Was it a member of a completely extinct kingdom, a fifth kingdom of life that existed only in the Ediacaran and died out entirely before the Cambrian? This was the proposal of the paleontologist Adolf Seilaker, who called the Ediacaran organisms Vendobionta and argued they were not animals, plants, fungi, or anything else, but a genuinely separate experiment in multicellular life. Was it an early animal? Specifically an early relative of bilaterians, the group that includes all animals with bilateral symmetry. For decades, there was no way to settle this because molecular evidence from soft-bodied Ediacaran organisms seemed impossible to obtain. The 2018 resolution, and its limits. In 2018, a paper in Science reported what appeared to be a genuine breakthrough. A team led by Johann Brox at the Australian National University analysed specimens of Dickinsonia found in Russia, preserved in ancient rock where the original organic material was retained. They identified cholesterol molecules, specifically cholesterol in the form found only in animals, in the Dickinsonia specimens. This strongly suggested Dickinsonia was an animal. And it placed the origin of animal multicellularity significantly earlier than previously confirmed. But, and this is crucial, identifying it as an animal doesn't identify what kind of animal. Dickinsonia has no clearly homologous structure to any known animal phylum. It has no identifiable nervous system remnants, no digestive system remnants, no circulatory system remnants. If it's an animal with a body plan so different from anything alive today, or anything known from the post-Cambrian fossil record, that we cannot determine what group it belongs to. It may represent a lineage of animals that evolved, flourished globally for millions of years, and then went entirely extinct, leaving no descendants and no anatomical blueprint we can recognize in any living creature. The remains of Dickinsonia are among the most studied fossil impressions in the world. And they still match no known species or category of animal we understand. A historical mystery with modern resonance. This case is different from the others. It doesn't come from the ancient fossil record. It involves physical material collected within living memory, and it remains, to date, scientifically unresolved. On August 7, 1994, residents of Oakville, Washington, reported a gelatinous substance falling from the sky during a rainstorm. The substance fell not once but 6 times over a 3-week period. People who came into contact with it reported becoming ill, symptoms including fatigue, severe nausea, dizziness, and in some cases breathing difficulty. Animals exposed to the substance also became sick. Samples were collected. What analysis found, and didn't find? The Washington State Department of Health analyzed samples. Their initial finding was that the substance contained human white blood cells, specifically 2 types of cells consistent with immune system material. This raised immediate and disturbing questions. Rain does not contain human white blood cells. There is no atmospheric or meteorological process that would produce gelatinous matter full of human immune cells falling from the sky. A microbiologist named Tim Davis at the Ameriquest Technologies lab in Kennewick analyzed samples separately and reported finding 2 types of bacteria, neither of which was identified as a common environmental species. The Washington State Department of Ecology conducted further testing but reported that the samples they received had degraded before testing could be completed. The samples were eventually all consumed in testing. No archival material remains. The analysis was never replicated. The Washington State Health Department's full report was never made publicly available in complete form. The matter was officially attributed, without strong evidence, to the U.S. military, which was conducting flight exercises in the area at the time, specifically, the bombing of jellyfish as part of a fisheries management operation. The theory, jellyfish tissue was aerosolized and fell as rain. This explanation has significant problems. Aerosolized jellyfish tissue would not contain intact human white blood cells. The geographic dispersion and volume of material is inconsistent with that mechanism. And no official military documentation of such an operation during that period has been made public. The broader pattern. The Oakville blobs are part of a broader phenomenon recorded for centuries, gelatinous masses appearing suddenly, on the ground or falling from the sky, with no clear origin. The Welsh called it putracere. Rot of the stars. It appears in medieval texts across Europe and Asia. Modern analyses of various samples collected globally have produced inconsistent results, some showing no biological material, some showing algae, some showing slime mold, some showing animal cells. No single species identification has been established. The phenomenon recurs. And we do not have an adequate scientific explanation for what produces it or what it is. This is a different kind of doesn't match any known species, not a fossil we're struggling to classify, but physical material, contemporary, falling from the sky, that analysis has failed to categorize. A new discovery with old implications. In 2019, paleontologists from the Royal Ontario Museum and the University of Toronto published a paper in Proceedings of the Royal Society describing a newly identified Cambrian predator from the Burgess Shale, Camberastor falcatus. The creature was named partly for its resemblance to the Millennium Falcon. Its head shield was wide, flat, and circular in a way that genuinely did look like the famous spacecraft. It was approximately 30 centimeters long, with frontal appendages for grasping prey and the characteristic oral cone of the radiodont group. What made Camberastor significant wasn't just the new discovery, it was the abundance. The site where it was found, called Marble Canyon, yielded hundreds of specimens. Camberastor was not a rare creature. It was common. The ecological problem. This creates a problem that paleontologists call the predator hierarchy problem of the Cambrian. The Cambrian ecosystem, as we now understand it, was dominated by large radiodiontan predators, Anomalocaris and its relatives. These were the apex predators. But apex predators, by definition, cannot be numerically dominant. An ecosystem cannot sustain large numbers of top-level predators without a pyramid of prey beneath them. The math doesn't work if apex predators are the most abundant organisms. The discovery of Camberastor in large numbers, alongside continued discoveries of other radiodont species, suggests that the Cambrian ecosystem was far more complex than our current models accommodate. These creatures may not all have been apex predators, some may have been filter feeders or mid-level predators. But here's the unresolved part: we cannot adequately reconstruct the Cambrian food web. We don't know what most of these creatures ate, how they competed, how the ecosystem was structured. The body plans are so different from modern ecosystems that applying modern ecological models produces contradictions. Camberastor's remains were found in 2012 but only formally described in 2019. Analysis is ongoing. Its exact diet, behavior, and ecological role are genuinely unknown. It is a creature whose physical remains we have in abundance. And we cannot tell you what it did or how it fit into the world it lived in, because that world had no modern equivalent. A Peruvian cemetery and an uncomfortable discovery. In 1928, the Peruvian archaeologist Julio Teo excavated a massive burial site on the Paracas Peninsula in southern Peru. He found over 300 mummified remains, in remarkable preservation due to the extreme aridity of the region. Many of these remains had skulls of unusual elongation. The skulls were significantly longer than normal human skulls, not just at the back, but in overall volume. Teo attributed this to intentional cranial deformation, a practice known from many ancient cultures where infants' heads are bound during development, reshaping the skull as it grows. This explanation was generally accepted for decades. What later analysis complicated? The problem is the parietal plates. A normal human skull has two parietal plates, the bones that form the top and sides of the skull. These are separated at birth and fuse as we age. Many of the Paracas skulls have a single parietal plate. Intentional cranial deformation, head binding, can change the shape of a skull. It works by applying pressure during the period when the skull is still developing and the plates haven't fully fused. It cannot change the number of plates. The number of parietal plates is genetically determined. Skulls with one parietal plate instead of two represent an anatomical variant that is not documented in modern human populations at any significant frequency, and is not documented in the anthropological record as a normal variant. Additionally, the overall cranial volume of some Paracas skulls has been measured at up to 2.5 times that of a modern human skull. Cranial deformation changes shape, not volume. You cannot bind a skull into having more interior space than it started with. Volume is fixed by the amount of brain tissue growing inside. DNA testing and its results. In 2014, the director of the Paracas History Museum, Brian Forster, arranged for samples from 5 of the Paracas skulls to be sent to a geneticist. Identified publicly only as Lakota, for mitochondrial DNA analysis. The results, as reported by Forster, showed haplogroups consistent with European and Middle Eastern origins, not consistent with pre-Columbian South American populations. This finding has not been replicated in a peer-reviewed context. The geneticist's identity and institutional affiliation have not been formally published. The methodology has not been independently verified. Peru's Ministry of Culture has periodically announced formal studies of the Paracas skulls, but comprehensive, peer-reviewed genetic and morphological results have not been published in major scientific journals. What we are left with. The Paracas skulls remain in a frustrating state of partial knowledge. The single parietal plate characteristic has been observed by multiple researchers. Its significance is disputed. Some argue it may be an artifact of post-mortem bone fusion, not a genuine in-life characteristic. But this has not been formally studied and published at scale. The cranial volume measurements, if accurate, are inconsistent with cranial deformation of otherwise normal human skulls. The genetic data that exists is methodologically uncertain. We have over 300 mummified remains from a single burial site. Some of them show anatomical characteristics that don't match the known morphological range of any documented human population, ancient or modern. They may be human. They may be extreme examples of genetic variation within our species. They may represent a population with unusual morphological characteristics that has not been studied with modern methods. But right now, we cannot tell you definitively what they are. And the institutions that have physical access to the remains have not published the kind of comprehensive, peer-reviewed analysis that would settle the question. The remains exist. The question remains open. 10 sets of remains. 10 creatures, or things that might be creatures, that don't fit. Some of these are ancient beyond imagination. 500 million years old, locked in rock, studied by some of the best paleontologists who ever lived. Some are recent. A 1994 gelatinous rain. Mummies from a century-old excavation. Some have been analyzed with the most sophisticated tools modern science has available, DNA sequencers, electron microscopes, synchrotron radiation tomography. And all of them, in their different ways, produce the same result: we don't have a complete, verified answer. There's a tendency to think that science has mapped the tree of life. That we know what has lived on this planet and roughly how it all fits together. The cases we've covered tonight suggest that picture is radically incomplete. The deep ocean, covering 71% of Earth's surface, has been explored less thoroughly than the surface of Mars. We have recovered fossils from perhaps a fraction of a percent of the species that have ever lived. The Cambrian explosion alone may have produced hundreds of animal body plans, most of which left no living descendants and only fragmentary remains. The honest position, the scientifically accurate position, is that we know far less than we often present ourselves as knowing. And that's not a failure. That's an invitation. Every one of these creatures represents a door. Behind each one is either an explanation we haven't found yet, or something genuinely new about the history of life on this planet. Science doesn't close doors. It opens them and follows them down. That's what makes this worth paying attention to.